In the next few lectures, you will be learning about the fundamental processes in molecular biology that determine how cells pass information to their offspring, and how that information determines how a cell will function. At the core of these processes are nucleic acids. This video will review the basics of nucleic acid structure and function. We will first look at nucleic acid function. One type of nucleic acid is DNA. It is essentially the blueprint for a cell in that it contains the precise instructions needed to make all necessary proteins. Specifically, DNA spells out the amino acid sequence for a protein. Remember from earlier in the course that proteins are the workforce of the cell and determine a cell's function. An important property of DNA is that it is able to be copied. This allows all of the protein-making information to be passed from parent to offspring cells. The second type of nucleic acid is RNA. There are many different kinds of RNA, a few of which are involved in protein synthesis. One such type is messenger RNA, abbreviated mRNA, and carries information from DNA to the protein-making machinery in the cytosol. All nucleic acids are polymers of individual subunits called nucleotides. Let's take a look at how the nucleotides of DNA and RNA differ. All nucleotides have a nitrogen-containing base. A nucleotide of DNA has either adenine, guanine, thymine, or cytosine. A nucleotide of RNA has adenine, guanine, and cytosine, but can contain uracil instead of thymine. Adenine and guanine are purines and are larger bases than the pyrimidines thymine, cytosine, and uracil. All nucleotides contain a 5-carbon sugar. In DNA, the sugar is deoxyribose, and in RNA, the sugar is ribose. The two differ by a single oxygen atom. Both DNA and RNA nucleotides contain a phosphate group. Lastly, the nitrogenous bases can form hydrogen bonds with each other in a process called base pairing. In DNA, adenine pairs with thymine, and guanine pairs with cytosine. In RNA, adenine pairs with uracil, and guanine pairs with cytosine. Here you can see all of the components of a nucleotide. The 5 carbons of the sugar are always labeled in a clockwise manner as shown. The nitrogenous base attaches to carbon 1, and the phosphate group attaches to carbon 5. When nucleotides bond to form a chain, the phosphates attach to the 3rd carbon of the previous nucleotide. In a nucleic acid chain, the end with the 5th carbon exposed is the 5' end, and the end with the 3rd carbon exposed is the 3' end. This directionality will be important to remember when we discuss replication, transcription, and translation later. DNA is actually composed of 2 chains of nucleotides, and the 2 strands are anti-parallel to each other. This means that looking at the molecule from top to bottom, for example, one strand has its 5' end at the top, and the other has the 5' end at the bottom. This anti-parallel nature gives rise to DNA's double helical structure. Covalent bonds connect the phosphate groups to the sugars, and this forms the backbone of the molecule. Hydrogen bonds connect the bases of the 2 strands. AT bonds are weaker and have only 2 hydrogen bonds. GC bonds are stronger and are held together by 3 hydrogen bonds.